Hello friends, welcome back to my YouTube channel BioWallah. In this video, we will be going to discuss about how monoclonal antibodies are being produced by Hybridoma Technology and what is Hybridoma Technology. So, George Collard and the Cecil Milstein invented the Hybridoma Technology in the year 1975 and they got Nobel Prize in 1984 for their discovery of hybridoma technology. Now let's see. So here, this is antigen and this is antibody. So antigen say here you can see epitope is attached on antigen and here paratope which is bind on antibody. When antigen and antibodies get bind, it will form antigen antibody interactions. So as you can see in this diagram, antigen antibody, they get attached, they get bind and they form antigen antibody interaction. So what is antigen? Antigen, a substance that binds with a specific antibody. So antigenic substance which may be bacteria, which may be pathogen, कुछ भी हो सकता है. So antigen एक substance है जो कि bind होता है specific antibody से. Now let's see the epitope. An epitope, also known as antigenic determinant. In this diagram, this is epitope which is bind with antigen. An epitope which is also known as antigenic determinant, and epitopes are the immunologically active region on a complex antigen that is recognized by immune system, specifically by antibody, B cells and T cells. Now antibody, so this is the antibody जिससे paratope which is attached with antibody. So antibody which is denoted with the A and B. So antibody also known as immunoglobulin, short form में IG से उसे denote करते हैं. So antibody को ही immunoglobulin कहते हैं. It is a large Y-shaped protein that the immune system utilizes to recognize and destroy foreign substance like harmful bacteria and viruses. The pathogen distinctive molecule known as antigen is recognized by antibody. So paratope, the paratope is a part of antibody which recognized an antigen. The antigen binding site of antibody. So paratope is nothing but the antigen binding site which is present on antibody. So this is the paratope जो क्या करेगा, एंटीजन को बाइंड करेगा एंटीबॉडी से. Now so let's see the monoclonal antibody. Monoclonal antibodies derived from single clone of cells and thus specific for a single epitope. Now, as I told you that hybridoma technology, a technique which produces monoclonal antibody. So, hybridoma technology is one of the most common method used to produce monoclonal antibodies. Hybridoma technology is a method of forming hybrid cell lines. We call them hybridoma by fusing a specific antibody producing B cell with myeloma cell, which is cancerous B cell. The antibody produced by hybridoma are of single specificity, that is, it is specific for single epitope, or this antibody is specific for only antigen of interest, only for a single antigen. Now B cells generally die within a few days in vitro. Hence, fusing them with myeloma cells gives an immortal cell line. Now, let's discuss and let's see the hybridoma technology. Now, here, this is a mouse which is challenged with antigen. Here, we have to develop an antibody which is highly specific to the our antigen of interest. What we do, we immunize the antigen of interest into the mice. What we do, we will be immunize the antigen of interest into the mice, and during the next specific week, the antigen specific B cells proliferate. So these are the B cells, which is antigen producing B cells. Now, this antigen producing B cell get fused with myeloma cell. So it will form hybridoma. So this is nothing but a fusion of antigen producing B cell and myeloma cell. Now antibody producing from this cell will be vary from the antibody producing this cell. So this cell will form a different antibodies. This cell will form a different antibody. This cell will form a different body, a different antibody. But let's suppose this is the cell. Let's suppose this is the cell which will be produced antibody which is specific for our antigen of interest. तो सपोज कि ये सेल है जो कि एंटीबॉडीज बनाएगा हमारे एंटीजन ऑफ इंटरेस्ट के लिए. So what we do here, we take this cell in a culture, we culture that cell in a hat medium. We culture that cell in a hat medium, and hat medium generally contain hypoxanthine, aminopterin, and thymidine. So we took this cell and we culture this cell into the hat medium. So we culture this cell into the hat medium, and after that this cell will produce antibodies which is highly specific for our antigen of interest. That's why we call them a monoclonal antibody, because these antibodies are highly specific for the antigen of interest. ये जो antibodies है सिर्फ इसी के लिए specific है. This is not specific with this antigen and this is not specific with this antigen. So ये जो antibody है ये इन दोनों antigen के लिए specific नहीं रहेगा. ये antibody सिर्फ और सिर्फ इसी antigen के लिए specific रहेगा. That's why we call them monoclonal antibody. Now let's see the applications of monoclonal antibodies. So the applications of monoclonal antibodies. Monoclonal antibodies are used for diagnosis, disease treatment, and research. Monoclonal antibody widely used for cancer treatment, where monoclonal antibodies used as a drug assist the natural immune system functions in fighting cancer. Thank you for watching. For more such videos, please subscribe to the channel BioWallah. If you like the video, then don't forget to share with your friends.